Alright, so let's talk about DNA replication. As we know, DNA replication should be happening during the S phase of interphase within our cells. And it's going to take place in two different steps. So the first step is your DNA is going to be unwound by an enzymatic process catalyzed by the enzyme helicase. So helicase, what it does is it's going to break the hydrogen bonds between your nitrogenous bases that dangle down the center of your DNA, creating this bubble. It opens up your DNA, unwinds it so that you have two single-stranded pieces of DNA that are exposed to things like enzymes, such as DNA polymerase. So helicase unzips your DNA, it unwinds your DNA, breaking those hydrogen bonds. And what happens is DNA polymerase, so polymerase ends in an ase, it's an enzyme. What does it do? Probably makes a polymer of something. What is it making a polymer of? DNA. So DNA polymerase comes along and it's going to add free nucleotides to the 3' end of a new strand. Okay, so DNA can only be written in a 5' to 3' direction. So what's going to happen is the mother strand or this template strand, you're going to have a 3', so it's going to have a 3' in this direction, and it's going to move towards a 5' at this end. Now remember, our DNA is anti-parallel, so as we write our DNA in the 5' to 3' direction, you have your new strand, which is going to be 5' over here, and it's going to be adding nucleotides to the 3' end as helicase unzips your DNA. So as you notice, this is happening on both of our strands, right? We call this semi-conservative replication. So one strand is being used as a template for one new strand, while simultaneously the underside of that DNA, of that same mother DNA, is going to be used as a template for another new strand of DNA. Now remember, they're anti-parallel, so they're actually going to be writing in two different directions. So we're just looking at the top here. This is called the leading strand. It's pretty easy for the leading strand to write DNA, or for DNA polymerase to write in this direction, because helicase is unzipping your DNA in this direction to the left, and our DNA polymerase, all it has to do is just follow along helicase, and as helicase opens up that DNA, DNA polymerase can make that polymer of DNA. So it can use that mother strand as a template to create new DNA. Now, also keep in mind is when DNA is being replicated, typically what you're going to do is you're going to unzip some region of your DNA, and you're actually going to have helicases running in both directions. So it's going to be running to the right and to the left. In this case, we're just looking at it running to the left, but the same thing is going to be happening on the right side. So just keep that in mind. So anyways, the leading strand follows along helicase, writing in a 5' to 3' direction. So DNA unwinds via helicase, and then DNA polymerase is going to add free nucleotides to the 3' end of the new strand. So the leading strand is pretty easy. It's pretty straightforward. It just adds nucleotides to the 3' end as helicase opens up the DNA. Now, the bottom strand, we call that the lagging strand. So it's actually a lot more complicated. So primase, actually before we get on to the primase, let's just look at this figure now. It's pretty much the same thing. So remember, you're writing in a 5', so here's your 5'. Let me get a laser pointer. So we got a 5' end over here, and it's writing towards the 3' direction, so 5' to 3' direction. So again, this is the leading strand. This square-shaped gray thing is DNA polymerase, and it's just pretty much following the helicases that are opening up the DNA. Okay. Now down below we have the lagging strand. Remember, we can only write in a 5' to 3' direction, and so you're going to have to write in that direction. You can't write it chasing your helicases. You have to write it in the opposite direction. And so what happens is you get this enzyme called primase, ends in an ase, probably an enzyme. What is it doing? It's probably putting a primer of something onto something. So primase comes along and it adds this RNA primer onto the template strand of DNA so that your DNA polymerase can bind to it and begin writing. So here we have primase, which is depicted by this teal square, and then the RNA primer is actually this magenta colored fragment. And your RNA primers are about roughly 10 nucleotides long. So your DNA polymerase will bind to that, it'll start at that primer of RNA, and it'll begin writing new DNA down in the 5' to 3' direction. Now, the beginning part, you would say that, like this was probably created, and then as it opened it up further, as helicase was unzipping further, then you had to create this fragment, and then you had to create this fragment, and then you had to create this fragment. And so like you would have like, as it opens it up further over here, you'd have to create another fragment leading in this direction. All right, so RNA polymerase writes its DNA up until it reaches the next, what we would call an Okazaki fragment or Okazaki fragment. And it's named that because the person who discovered this, his last name was Okazaki or Okazaki. So you go from RNA to DNA, which meets RNA to DNA, and so on. So what happens is after this DNA polymerase meets this RNA, it'll just disassociate. It'll dislodge from this strand of DNA, and what will happen is another RNA polymerase, or sorry, not RNA polymerase, DNA polymerase will come along, remove the RNA, and add DNA to it. Or not add DNA to it, but it'll replace the RNA with DNA. Because remember, we're trying to duplicate our DNA with 100% DNA. We don't want a mixture of DNA and RNA. So another DNA polymerase will come and replace the RNA with DNA. However, in doing that, you actually create a little gap in the backbone of the DNA. And so another step is DNA ligase. It's an enzyme. Can't really infer what it does based off of the lig part of it. But DNA ligase will come along and it'll close up that little opening in your DNA. So yeah, after the RNA primer is replaced by DNA, DNA ligase joins the Okazaki fragments to the growing strand. So again, your lagging strand is a little bit more complicated. It involves more enzymes and different enzymes. Okay, so what's typically going to happen as you are replicating DNA is sometimes you'll make some mistakes. And as you make those mistakes, well, what happens is your DNA polymerase is actually kind of proofreading as it goes. So proofreading is conducted by DNA polymerase, and as it makes a mistake, it's corrected before it continues. Now, if it doesn't catch its mistake, what happens is typically still in S phase, you're going to get a process called mismatch repair. So immediately after replication, a system of enzymes will scan for the mismatches and then repair them. So that's going to be conducted by a set of enzymes. So this on the left is actually what's depicting mismatch repair. So a mismatch is detected in newly synthesized DNA. The DNA strand is cut out, and then it is repaired by an enzymatic process, and then ligase will come and seal the gap of that backbone. Now, after your cell has replicated its DNA and it's gone through S phase and G2 and maybe meiosis or mitosis, and then maybe it's like in G1 or G0 phase, it might accrue damage, and you actually have a different system of repair during a different period in the cell's life. So it's going to also use different enzymes. And typically, well, we would call this excision repair. And typically what excision repair is looking for is structural changes to DNA that would that would disrupt the helical shape of DNA. And so in this case, it uses a thymine dimer as an example. So you have a thymine bound to a thymine, not making those hydrogen bonds with their adenine counterpart or the complementary strand, and they make maybe like a covalent bond between the two different thymines. And so that would typically disrupt the helical shape of the DNA, and that would be detected by a set of enzymes. And what would happen is you would create a bubble, you'd open up the DNA and separate the two strands of DNA, you'd excise that region with the error, or with the, in this case, the thymine dimer, and then you would have another set of enzymes come and repair the damage, and then ligase would seal the backbone after that. So mismatch and repair and excision repair, pretty similar processes, though orchestrated by different sets of enzymes, and it's going to typically happen at different times in the cell's life. So mismatch repair is going to typically happen during S phase, whereas excision repair can happen during any phase. And then again, proofreading is something that's going to be conducted by DNA polymerase as it continues to duplicate DNA or create that polymer new strand of DNA. So the DNA polymerase error rate is about 1 in 100,000, which would amount to 120,000 mutations for every cell division. So that's a lot of mistakes within just one cell division. However, because of proofreading and mismatch repair, the error rate is reduced to 1 in 10 billion. So last topic here is telomeres. So telomeres are repetitive sequences at the end of chromosomes that kind of act as a protective cap at the tips of your linear chromosomes. And their morphology, or what they're composed of, are these stretches of TTA, GGG, TTA, GGG, TTA, GGG, and so on, and it just kind of continues. And that's kind of like the, the protective region of your chromosome. However, they're not replicated fully with every cell division. So every time your cell divides, or not every time your cell divides, but every time you duplicate your DNA, you do not duplicate them with 100% of the telomere length. So typically, because we know that telomeres will shorten with age, we can often kind of infer the age of a cell based off of the length of its telomeres. So here we just have an example of, your origin cell, and it's got a large telomere, and then you have a division, slightly shorter, another division, slightly shorter. So mechanistically what happens is, if we're looking at, let's just look at this top strand, for example. You've got your 3' end over here, you have your 5' end over here, and remember, when we copy DNA, we're going to create a bubble in the middle of this DNA, and we're going to be writing in both directions. So in our examples earlier, we were just looking at the left side, but in this case, we're actually looking at both sides. So if we were writing in this direction, we'd be writing, from the 5' end of our new DNA in the direction of the 3' end, right? Because it's anti-parallel to the template strand. And so in this case, the leading strand would just follow helicase and write all the way to the end over here. However, on the right side of the origin of replication, this is actually the lagging strand now on the right side of the origin of replication. Because you remember, you have to write from 5 to 3, and so as helicase is opening up the DNA in this direction, you have to create these Okazaki fragments. Okazaki fragments kind of chasing that helicase. So when the replication fork eventually reaches the end of the chromosome, in many species, including humans, what's going to happen is you actually don't have enough space to put the final Okazaki fragment, because you don't have enough space to put that final primer at the very end. And so as a result, you actually get a little bit of overhang. So you fail to replicate this region right here onto the new strand, And so you get this little overhang of the original strand here on the, you could say, the lagging end of it. So here, let's say the final segment of DNA, stops here at the G. Well, this is 1, 2, 3, 4, 5, 6, 7 nucleotides long. You can't fit a primer and an Okazaki fragment right here. So therefore, you're just not going to replicate this area. And therefore, you get that overhang. And over time, what that leads to is the shortening of your telomeres.